Let us start our program with prayer song.
May the Lord bless you and keep you, make His face shine upon you. The Lord be gracious to you. May the Lord bless you and keep you, make His face shine upon you. The Lord be gracious to you. May His love and His grace and His perfect peace guard your heart and your mind and your life. May His love and His grace and His perfect peace guard your heart and your mind and your life. May the Lord bless you and keep you, make His face shine upon you. The Lord be gracious to you. May the Lord bless you and keep you, make His face shine upon you. The Lord be gracious to you. May His love and His
A very great morning to all. Let us commence our third day international webinar with, on behalf of the management of JMJ College for Women, Tenali, the Department of Chemistry, I extend a warm welcome to all for the third day of the international webinar on recent innovations in pharmaceutical and applied sciences. I, Dr. K. Nirmala Jyoti, HOD of Chemistry, would like to introduce today's eminent resource person, Dr. Susan Kumar, physical therapist from Canada. Dr. Susan Kumar Vadakoppala hails from Bapatla, Guntur district, AP, India. He completed his school studies from Holy Cross English Medium School, Bapatla. He received intermediate education from Salvation Army William Booth Junior College, Bapatla. He is an outstanding all-rounder. He is an avid sports person who participated in various sports like volleyball, cricket, mini marathon, athletics, caroms, chess, and table tennis. He also participated in many extracurricular activities like debate, elocution, dramatization from school level to college level. He won various prizes in mono action, quiz, Bible games, and dance. He also conducted Bible classes, outreach programs, encouraging children and youth to build good conduct. Dr. Susan Kumar graduated with Bachelor of Physiotherapy from NTR University, India in 2008. He worked for a neurosurgery clinic from 2009 to 2011 before moving to a super specialty hospital in 2011 until 2018. Over 7 years in the 600-bedded hospital, Susan worked in outpatient department, intensive care unit, and orthopedic post-surgery care. He gained experience in assessment, diagnosis, and treatment of conditions related to musculoskeletal, neuromusculoskeletal, and sports-related injuries. Later, he moved to Canada with family in 2018 as a permanent resident and worked for Good Samaritan Care Home as a rehabilitation assistant. Then, he joined as a team member for Dynamic Balance Physiotherapy Clinic as interim registered physiotherapist. Dr. Susan Kumar is diligent towards his work, shows empathy to his client and loves to see them go pain-free. Dr. Kumar, on his day off, gives his undivided attention to his family. We are truly honored and thankful, sir, for accepting our invitation despite of your hectic schedule. Now, moving on to today's session, which mainly focuses on lifestyle modifications. Lifestyle changes are behavior modifications or habit changes that encourage positive changes in your life. These are a fundamental component to any wellness program. Making changes to these habits can create lasting effects on your personal well being. The foods you eat, the amount of sleep you get, and your exercise habits can all influence your weight, hormone health, and level of pain. Good health is incredibly complex, and there are lots of tiny tweaks you can make to your daily habits in order to help you live longer and with more energy, focus, and happiness. Now, I cordially invite Dr. Susan Kumar, physical therapist from Canada, to enlighten the participants with your scintillating lecture on lifestyle modifications for better health and positive well being. Thank you, sir.
Thank you so much, madam. Dr. Nirmala Jyoti madam has covered most of the topic which I'm about to say on lifestyle modifications. I thank Dr. Nirmala Jyoti madam and JMJ College for giving me this opportunity. So getting into our presentation, just a second. Open my PPTs. I hope everyone can see the screen now.
No, sir.
Yeah. Okay. So lifestyle modifications and
Sir,
its impact on the health.
presentation రావట్లేదు సార్.
Presentation is
It's
not
not me. I'm sorry. I
Presentation is
think I
not
shared the screen. How can I do that one now?
Otherwise, share the window, I think.
Oh, window. How about now? Is it coming now?
ఓకే సార్.
Yes.
Yes, sir, it is come.
Yeah, now it's good, right? So okay, I cannot see myself now. I can only see my screen.
Your
So,
screen, but you see in bias, no problem.
Yeah, that's okay, right? Lifestyle and modifications of lifestyle and its impact on health. See how our habits change our life. So before starting this one, I want to say something like how our ancestors, our grandparents and great-grandparents lived. So once if we recollect those days, most of our grandparents and parents, they lived such a small in a small circle where they know their community very well, and the lifestyle, the way they eat, the way they do their activities is very simple way, but it was very healthy. No, you don't see most of them going to hospitals. Even we as children, we haven't been to any hospital, stayed overnight or spent overnight in hospitals for taking medicines or anything like that, because most of our treatment comes through our food habits only. We eat a healthy food, but now when we compare to the children and our grandchildren, the way they eat, it has become all processed food. So we'll go through that one and then see how it has changed. So thank you for so much for inviting and giving this opportunity. So once we think how a meal looked like in those times and now, there is a vast change in that one. Sadly, what happened is like we have reached a point globally where we are now more sicker than any time in the history. Diabetes, obesity, cancer, heart disease, neurological diseases, all at unprecedented heights. Yet, this is the same time we are spending more amount on healthcare rather than anything else. We don't see spending much on agriculture. We see spending more towards the healthcare system. Human body is one of the most extraordinary healing and regenerating systems that nature has ever produced. Lifestyle diseases share risk factors similar to prolonged exposure to three modifiable lifestyle behaviors: smoking, unhealthy diet, and physical inactivity, and result in the development of chronic diseases, specifically heart disease, stroke, diabetes, obesity, metabolic syndrome, chronic obstructive pulmonary disease, and some types of cancer. Metabolic syndrome. Metabolic syndrome is something like where increased blood pressure, high blood sugar, excess body fat around the waist, and abnormal cholesterol levels, that all make up metabolic syndrome. So when we see our population, since we have exponentially grown in the numbers compared to the last 100 years, in last two to 3 decades, we have raised double. And then in future, in next 10 years, we are going to surpass every country in the world and be in the top on the population. So being that one, saying that, I'm not against the population, but again, it is like when we are in that numbers, we have to keep some habits or some lifestyle where everyone else can see and learn from us. So what happens is, when it comes to health, a cost of CABG, one heart transplant or heart surgery, is costing around 95,000 to 4.5 lakhs in India. So when it comes to diabetes, a treatment for diabetes for lifetime is costing minimum of 10 lakhs to 28 lakhs. Everything is going towards the private sector, not public sector. No one is going towards the government hospitals for treatment. Everything is happening in the private sectors. So when it comes to stroke, a neurological disease where paralysis happens in that condition, the treatment for lifetime is costing around 32 lakhs. The annual cost of treating the consequences of a heart disease, diabetes, liver disease, any depression type of diseases, is any types of cancer, is going to reach a staggering amount of 13 crores in India by 2025. That's just another 3 years, not much far away. So the global annual medical cost for treating a serious consequences of obesity is expected to reach 1.2 trillions by 2025. That's from the World Obesity Federation data. So what's happening is, this is the heart cardiovascular diseases. The number of deaths happened from 2010 to 2021. In just 10 years, it has increased so much. And then this one is the behavioral risk factors that causes cardiovascular disease due to dietary risks, tobacco use, and low physical activity. India stands in that orange, where we are always high in tobacco consumption and then low physical activity from 1990 compared to 2016. The blue one is globally, whereas orange is India, and then compared to United States, we are always in the high level of having low physical activity or dietary risks. So failures in the pharmaceutical regulatory environment in India have contributed to oversupply of ease and access to various medications. Lots of medications, where including with many with the little evidence to support the safe usage. This combined with poor community literacy about medication safety. We are not well trained about the side effects of medications at all. And usage and potential misuse and overuse of medication in India, this in turn can contribute to ill health, public health predicaments such as antibiotic resistance. We are losing that resistance in our body, which we had so much earlier. And further impoverishment of the community. Due to poor and non-existent regulations, it has been estimated that at least 50% of the average family spending on medicines in the country is incurred to irrational and unnecessary drugs and diagnostic tests. We spend a lot of money which is not necessary. So the pills and procedures that have been a gold standard for maintaining and improving the health have failed. What is the main usage of having a medicine? To improve our health. But on the opposite side, it's causing adverse effects on health. So is it wise to use medicines? Is it wise to take pills? So what happened is, when it comes to medicine usage, in 2020 study, a study found out Indians consume significantly more calories than the recommended diet, whereas the consumption of fruits is very, very low. Vegetables, meat, fish, and eggs are much lower. Protein share was only 6 to 8% compared to the 29% of recommendation. 29% was recommended in a meal, but we end up taking only 6 to 8%. These outcomes were independent of the socioeconomic status. It is not like rich are having very good food and poor is not having good food in Indian households. Even the rich turned out to be, in this study, were not found to consume optimum amounts of fruits, vegetables, and proteins in their diet. In fact, an Indian household consumer more calories from processed food. Authors concluded average Indian diet is unhealthy, and then lack of essential nutrients. It doesn't have sufficient nutrients to support. So what is going to happen to the next generation if it is going to be all unhealthy food around? There must be a better way, right, for all this. Even the children, when it comes to children always, they do not follow whatever we say to them. They are like what we can say is like poor listeners, but they never they never stop to imitate us. They never miss it. Always, whatever we do, they do it. If we end up doing like this, they follow us automatically. Many smokers might say, so I am not at all, only smokers are sick, whereas a non-smoker can say, I'm not at all worried because it's they who are spoiling their health and others' health. But it turned out in a research, even the person who sits 8 to 10 hours or spends most of the time sitting is a chronic smoker. The new disease is sitting. We end up sitting most of the time. Today I decided to stand up and take the presentation the whole time, whatever is going to happen, thinking like it is a change. So even if we take that small decision of standing up or doing some activity while doing, say for example, you are heating up a cup of coffee, doing some sit-ups there. Just 10 sit-ups. It is enough for your heart to pump effectively. So that's what there are many kind of researches like that. So sitting is one of the smoking habit. It is equal to smoking. That's what researchers have said. So now what happens is like there must be a better way to nurture well-being and reverse the disease. That's what we have to look into, because we have already got so much sick sick everywhere. So we need to find a way of doing reverse. So what happened is like being in physiotherapy for over a decade and having the background from the family of not using much medication. You don't see much medication back in my home, but whereas people have lots of a complete a box full of medicines. You say like I have an acid problem, they will give you an antacid. They say, if you say like I have some cough, they'll have a cough medicine. Antibiotics are free, very easily available in regular households. So me being in the background of physiotherapy, what has happened was I treat patients from young age to old age. Whoever come to us end up saying, we come to physiotherapy because we are tired of taking medicine. We have tried everything. They try whatever doctors say, like take this medicine for that pain, take this medicine for this condition. And then they come back and then they try to seek a treatment or something away from the medicine pill usage. They don't like to take the medicine, or they must be tired. They have side effects of medicine. They realized it. So listening to that over the years, it has come to my mind that something is wrong with the medicines, or something is wrong. I'm not saying do not take it at all, but it is not doing what it's supposed to do. It is doing entirely opposite way. So there should be something. So I came across an article. It is in 2009. It evaluated 4 simple lifestyle factors. BMI less than 30 is not obese. And then do they smoke? No. And then do they participate 3.5 hours of walking? That is at least in a week, that is like 30 minutes a day, not even much. 30 minutes a day is like morning 15, evening 15 minutes of walk. Do they have diet that consists predominantly of fruits, vegetables, whole grains with low meat consumption? This is the only factors, the 4 simple lifestyle factors. When it comes to vegetarian, I'm not, no meat, I'm not saying completely vegetarian, not vegetarian, unprocessed plant-based diet. Plant-based diet is only like the one fruits, anything, say any vegetable which you name, that is just a plant-based diet. What was found was shocking after this study in 2009. People who could actually follow these 4 lifestyles, they were rewarded with a decrease in risk of disease, any chronic disease, by 80%. That's mind-blowing, right? 80%. If there is a pill that can do with this efficacy, 80% reduction of chronic diseases, every doctor would prescribe it for everyone above 30 years of age. Do we have anything such in our pharmaceutical or surgical armamentarium? Nothing that comes close to decreasing the risk of any chronic disease by 80%. For me, that is a bumper offer. We have something in our in front of us that with 4 lifestyle factors, we can decrease our diseases by 80%. That saves you from lifetime misery of dealing with chronic diseases, no heart diseases, no diabetes, no cancer, innumerable number of pills. That is the most thing going through our digestive system, ruining our body. We can save ourselves from all this. Now the issue is, how many of us are willing to go along with the 4 simple lifestyle factors that can indefinitely give a healthy longevity? How many of us? Less than 5%. Why is it so? Why, when we have everything in front of us, all very simple lifestyle factors, need not to invest much, need not to spend so much, but why do we end up only with 5%? When we know lifestyle factors have such dramatic impact of health outcomes, why do we have such dismally low percentage of people following it, actively engaging in these 4 healthy lifestyles? Do you know why? It turns out lifestyle choices are contagious. In a similar way to virus, so we can likely pursue like same type of habits. What happens is like we can likely to pursue same type of habits and activities those who are doing around us. For example, if you hang out with a person who likes to go for evening walks, and share only plant-based food, you are more likely to end up doing the same, following them. In the opposite way, conversely, if you hang out with people who spend a lot of time in front of screens, indulging on junky and sugary foods, not prioritizing enough sleep, you are less likely to focus on healthy eating and exercises and sleep. This is the power of social nudges. Social nudges is something where we look around and try to follow that, which means most of the people in the group follow along the rest of the group in doing it so. So what we should do? Do it tomorrow? That's what we postpone it always. So what happens is that research has showed healthy living is the best revenge. You live healthy, you are taking revenge on everything that is happening around. In 2000, scientists discovered 5 places in the world where people live the longest. They not only live long, but remain healthy, active, and vital members of their communities while spending very little on healthcare. Finding such places is a dream for many. So researchers started. They started analyzing the place, studying people, food, what they eat, the water sources, thinking that's a magical fountain so that they can bring back to their country and feed their people and suddenly become young. They studied their culture, how they live, what they do, like what is their lifestyle like. And the outcome was not something you can turn into a supplement or some pharmaceutical medicine or something like that. But the intention was for them to find out something secret happening there and then bring it to their countries and implement with their within their communities so that they can develop some secret medicine. But it turns out that it is not like that. Instead, what they found out was lifestyle choices are the social fabric of these communities. They are very closely knit, which we don't see in many other places. And these can be distilled down to the way you move, what you eat, how you handle stress, whether or not you feel a sense of belonging. That's the word most of the people looking forward is sense of belonging. So this practice in the social nudges and social architecture in this place, where blue zones lifestyle is followed, will have a default of lowering the risk of chronic diseases and increasing the habits that improve overall well-being. Just following those simple lifestyle choices. Are we going to follow this and create a blue zone in our communities? With such population, it should be possible. Are we going to follow this one? Maybe if we all bring awareness of this particular lifestyle within our communities, we could definitely. In fact, I believe deep within our roots, once we had this lifestyle, our ancestors had this lifestyle. Even before globalization, where villages and towns are remotely located, our ancestors were pioneers of this way of living. Only thing we have forgotten while following all this globalization or the Western culture or whatever it is, we have lost it. Once a famous cardiac surgeon said, the secret of a healthy life is eat what your ancestors ate in limit quantities, like vegetables, and have very simple, uncomplicated social life. That's enough. You don't have to do anything. You don't have to build up six packs, 6 abs, like do lots of time, spend lots of time in the gym or do activities. Very simply, he said. That's true. Heart disease is most likely to kill anyone out there here. Even in this webinar, in this group, heart disease can kill anyone. It doesn't see age or sex. We know healthy diet, regular physical activity, stress management, restorative sleep, social connections, avoiding tobacco, like all these things are preventive strategies. But what if you already have a cardiac disease? This research shows that one. If you already have a cardiac disease, it turns out that people who have severe cardiac disease put on low-fat whole food plant-based diet, just plant-based diet, can show increases in blood flow, makes their symptoms disappear. How long does it take following this diet? How long does it take for their symptoms to disappear? 3 weeks. In just 3 weeks, if you only stay on plant-based diet, your symptoms are going to come down. That's a good thing. Cardiac patients, heart trouble, heart diseases, people are spending years together struggling, finding medicines, taking an innumerable number of pills, not knowing what to do it. But here the research says within 3 weeks, it is possible. People who have been told that they were out of options had significant improvement in their symptoms in just 3 weeks. How about diabetes? Diabetes is the second most chronic diseases that's troubling people. In just 16 days, 16 days of having a whole plant food-based diet, again, plant-based diet, for the bodies to transform and begin the healing process. Good news is all the research is done, evidence-based research. It is right in front of us. All the experiments are done. Only thing is we need to follow it. We can tackle our chronic disease right now with tools that we already know is effective. They have done the research, they tried it on people, and it is effective. It is effective, evidence-based here. Food, physical activity, social connection, restorative sleep. We can create our neighborhood, our schools, colleges, and our workplaces to nudge us into these lifestyle choices that nourish us instead of causing us damage. They don't do damage. The power to achieve a long, healthy span and a long life that is full of vitality and purpose is really within our own hands. The power is in the relationship we nurture, the food we eat, the way we move and sleep, the culture we create. We can make the healthy choice the easy choice, both for us and for our children, their children. So the choice is ours and the impact will be truly extraordinary. I want to play this one video. Please let me know if the video is playing or not, if you guys can watch it. Just have a quick, can you guys hear it? Is the video playing?
Yeah, video is playing.
Okay, yeah. Just follow it for a few minutes. Thank you so much. Yes.
That's a really good one.
Yeah, thank you so much. It's just one choice, one thought, one idea. If we can spread it out and then learn it from that, definitely our life is going to change. Thank you so much.
Now I'm requesting the participants, if you have any queries, please ask. You can either type it in the chat box or you can unmute and you can ask.
Yeah, hi, everyone. I'm supporting Mr. Shushan Kumar. Yeah, what you in your presentation, what you said is standing and participating in anything is very good, because what you said is correct. Now in European countries and some other universities, they are still practicing that. There is no chairs in the classes. They will stand and do the work, because when you are stand, your mind will activate. When you are sit, your mind will relax. So at this standing and doing work is one of the good therapeutical exercise. When my mind should be activated, even in classes also. That's why in earlier days, teacher tell us whenever any little punish they give, but that is not the punishment actually. To focus it on the subject, please stand and you listen, like that they told that time. So many teachers explain like that. So that is very good one. So we have to cultivate in our daily life, that one.
Yes, yes.
Yeah,
That's true. What you said is really right. So what we believe here is any activity you do is gonna improve your health. It is okay. It is easy to relax and it is sometimes acceptable. But when you think in the other ways, like you have a few minutes to do it, it is better to do some activity. That's what they say. Like even a slightest stretch, just standing and then try to stretch yourself and then stand. It increases the blood circulation to different parts of the body. That's true. What you said.
Yeah. Little physiotherapy nowadays so much familiar. Now people are going to, but actually my presentation is little contradicted to your presentation, because mine is pharmaceuticals.
No, we are not against it because it is always
Yeah.
good.
But no. So yeah, what you said is we are also trying to do
Yeah.
to overcome the side effects. So
Side effects. That's the
by
word. What
introducing
you said is
new technology, we are
Yeah.
trying to impurities which are adverse effect to the human body. So we are goes on decreasing the ppb level, ppvm to develop the methods. So what are the best quality we have to give to the API? We are doing our best, and we are improving the technologies. So many regulatory bodies also keep on working. Pharmacovigilance people, they are getting data. What are the impurities? People giving side effects. So yeah. So my
There is always a
topic is,
balance, sir.
yeah.
There
So
is
but
always a
little
balance.
contradictory, but we are agree with you. What you
It
said
is
is
not
correct.
contrary. Yeah.
Yeah.
If anyone have any doubts, they can, if time is allowed. Whatever I'm trying to read the chat here. Gayatri Madam is saying, health is wealth. We are not carrying our health. Diabetes is very bad. So many numbers suffering from it. neglected healthcare. Medicine is one type of drugs. Thank you, ma'am, for your insights. No walking time, busy life.
Audience, please type the question in the chat box, or you can unmute
You
yourself.
can conclude. If there are no questions, I think you can conclude to the next section. Session.
Thank you, Susan Kumar garu, for your wonderful presentation. Thank you once again.
It's my privilege to propose vote of thanks. I would like to thank today's resource person, Dr. Susan Kumar Vadkupala, for his excellent and outstanding presentation. Sir, you enlightened us clearly about lifestyle, population, public versus private health care, heart diseases, risk factors, habits, and blue zones. Thank you all. We will continue next section.
Once again, thank you, sir, for your wonderful session. Thank you, sir.
You're welcome, madam.
Go to next session.
Now, I hand over to Purnima. Please carry on your session.
Thank you, ma'am, for giving me this opportunity. It's a great pleasure for me to introduce our today's resource person, Dr. Raja Gorla, who is going to give an invited talk on advanced techniques and applications in pharmaceuticals industry to achieve quality products. Dr. Raja Gorla is currently working as a senior scientist in Analytical Research Department of the Dongkwon Life Science Limited, South Korea. He has completed his PhD in chemistry from Andhra University and master's degree in organic chemistry from Ideal College, Kakinada, Andhra Pradesh. He is carrying almost 50 years of experience in the analytical research and development of pharmaceuticals industry. He has started his career as a research associate in the Aurobindo Pharmaceutical Limited Research Center, Hyderabad, India. There he has reached up to scientist level with good recognition. He has presented many papers in international and national seminars and published many articles in reputed journals. And two times he got awarded for the paper presentation in the International Symposium in Maharashtra and Andhra Pradesh. Sir, we are very happy to have you with us today. Before I hand over the session to Sir, I would like to read the significance of the day. As every aspect of the healthcare industry received global attention during the COVID-19 pandemic, the Indian pharmaceutical industry rose to this challenge. As when most sectors were brought to a halt due to the pandemic and the global lockdown, the pharmaceutical industry continued to strive and find a solution to the crisis. Our country exported essential drugs and medical equipments to more than 150 countries in the beginning of the pandemic, and then did the same with vaccination doses this year, which gained global recognition. Speaking about innovation in the pharma sector, its importance was reinforced during the pandemic, and the speed, scale, and willingness to innovate in the industry was impressive. Ignoring quality in pharma products is equal to risking humans' life and losing your credibility. Thus, quality assurance and quality control is important in pharmaceutical industry to manufacture and deliver complete zero defect products with excellent quality, efficacy, and safety for the consumers. By developing more efficient processes and switching from defect detection to defect prevention, we can improve the quality of the pharmaceutical products. With a thorough process of quality assurance testing, you can find out any potential bugs or issues that might occur in the future. Finally, by building quality into processes instead of improving the additional testing or inspections, one can achieve the quality of the pharmaceutical product. This is the subject in which we should all be deeply interested, and we have the right person with us to discuss. Now, I gladly invite Dr. Raja Gola, Senior Scientist, to take over the session.
Thank you, Purnama. Yeah, it's very wonderful. Very good session also. Yeah, good morning, all of you. I'm very excited to meet you all in this auspicious occasion.
Yes.
I'm very thankful to JMJ College Management and Department of Chemistry and Nirmala Jyoti ma'am and Shakina for inviting me and my presentation in this international webinar. Actually, recent innovations in pharmaceutical and applied sciences. So, actually, my topic is exactly apt with this this international webinar title.
Yes.
So I'm very happy. So my special thanks to all the people who are participated in this webinar, and please don't shy anybody. Please ask me questions after this completion of seminar, because I hope all our chemistry students, more chemistry students are participated in this webinar. So as a background from chemistry, so you have any doubts, so don't feel shy, please ask me. As an introductory part, so already they told, I have almost 15 years experience in analytical research and development, especially in pharmaceutical, active pharmaceutical ingredient. So all you know as a chemistry students, what is API? That is drug which are using in oral substances, and pills are like any creams. What are the formulation? There is a ingredient which is called as active pharma ingredient. So when you're going to the topic, I'm sharing my screen. Please go through it. I think it is visible, the screen. Oh, wait a second. Oh, wait a second. And I think it is visible now.
Yes, sir.
Correct. It is visible?
Yeah, yes, sir.
Yeah. Thank you. Yeah, students, let us start my topic. So all are ready? There is no response?
Yes, sir.
Yeah, okay.
Yes,
Today's my yeah. So today my topic is advanced techniques and applications in pharmaceutical industry to achieve quality products. So topic itself is clearly explaining that. So what are the current changes in pharmaceutical industry? What are the good manufacturing practices? What is GLP, good lab practices, to achieve quality products? Because earlier Shushan Mr. Shushan, what he said, so many diseases increasing, and recently we are aware this pandemic also we faced so many problems. So once, as Susan said, that the pills, what are the chemicals we are using, that all little side effects is there, but at last we have to solve the problem who is facing human beings in their daily life. They are facing so many problems. So up to that, they have some diseases which are struggling. So as a scientist, we are doing to overcome at least their problems to day-to-day life. They're giving some good quality products, good medicines. Uh, so, so goes to the topic. So, what are the present? If you go earlier days in almost 80s, 90s, that time we are using just simple classical methods like column chromatography, wet chemistry. So that time there is no that much what are the methods developed that time, classical methods. There is no result oriented. That means whether that result is correct, where it is reproducible, or some quality is 100% getting good quality. So we don't have any result oriented. That means this is correct or not, we don't know that time. But that time scientists also worked very well. So they goes on increasing every time the technology, how we have to improve our quality to overcome this reproducibility problem, sensitivity problems. So how you reduce the labor and how you fasten the work, how you give the productivity more. So they tried so much. So they based on technology and scientifically they added the technology and science, how technology will help the science to uh, improve the quality of the product and improve the productivity of the product. Because simply you are just synthesizing any product and it is not getting to all the patients, uh, it is not useful. So we have to improve good quality products and productivity is also more. So then only the technology what we are using, that is really helpful for the science and helpful for the human being. So as a instruments are revolutionarily have so many changes year to year. So because you know, so many regulations are there for it's not a simple pharmaceutical industry. It's not a simple industry. Just we are producing any product and sell. It is not. It's not easy because there are so many regulations. There is ICH guidelines, international harmonization to how to if you give any product, it is how do you give the product compliance, is it is in the compliance or not. So they check all, there are so many regulations are there. So for getting approval from that agencies, industries are working on how to give good quality product. So how do we get agreement? We get agreement from how we can easily sell our product overall the world. So how to become so much, because now industry is very competent. So people are trying to use technology to agreements with other countries. So to overcome to get good products. So like that, so technology has a revolutionary changes in chemical analysis. So replaces wet chemistry methods, higher sensitivity and accuracy, increased lab productivity due to automation. So if you see 21 CFR is nothing but a code for federal regulations for good manufacturing process and good lab practices and good manufacturing practices. So go through the pharmaceutical industry. So first separation techniques. So already I think this we have chemistry is divided into three parts. One is four in organic chemistry, inorganic chemistry, physical chemistry, and analytical chemistry. So if you go through the analytical chemistry, this separation techniques, you are so many analytical students aware of these techniques. High performance liquid chromatography, gas chromatography, HPTLC, and ion chromatography, mass spectroscopy, and so many life science instrumentation, sequencing, DNA synthesizer, organic and peptide synthesis. If you go through the spectroscopic techniques, so these are the new technology instruments now currently working in pharmaceutical industries. So atomic absorption, UV visible, ICP-OES, MS, fluorescence, infrared spectroscopy, electron microscope, light microscope, scanning, material characterization, thermal analysis, viscometry, and lab automation, information management, robotics, liquid handling, and microplate reading. So these are all new technologies now currently working in the pharmaceutical industry and currently using to give the best quality products. Just in this slide, I shown some of the instruments images which are currently. So maybe some of the students may be aware of this. This is HPLC waters, and this is HPLC Agilent, and this is HPLC Shimaju. So these three companies now familiar in pharmaceutical industries, and these are all good quality instruments which are following all regulatory bodies suggestions, and they day to day they are developing the technology to give good separations. So this is also one of the slide showing the diagram of this gas chromatography GC, and this is one Agilent manufacturer, and this is Shimaju, and this is LCMS. So for liquid chromatography, we attached some mass spectroscopy, and this is gas chromatography, which attached with the mass spectrometer. To go through this is nuclear magnetic resonance instrument, which are currently we are using in the pharmaceutical labs for characterization of the molecules. And this is XRD, which you are used for how the character of the physical characteristic of the material, whether it is amorphous or crystalline. So by this instrument we can characterize the physical characteristic, and this is characteristics by using NMR. We use the characteristic of the molecule to predict the characteristics of the molecules. And what are the techniques now I shown? So what are the applications by the techniques we are presently using in pharmaceuticals? If you go for HPLC, high performance liquid chromatography. So as a student, theoretically you learn daily, yeah, this is HPLC used for separation of compounds, and this is GC, gas chromatography. By using gas chromatography, we can analyze volatile components. So theoretically, we know at the PG level, post-graduation level, we have so much of books available for this separation techniques. So when you, as I know, after completion of my post-graduation and PhD, we just simply entered into the lab. We also, the theoretical part is okay, but when you go for the application part, we are, because lack of our infrastructure in the colleges, we don't have awareness of what is this instrument, because we theoretically we know. So this is what are the HPL, high performance liquid chromatography is used for separation of this organic compounds, and gas chromatography is used for quality. But how we analyze the samples, we don't know anything. Just as a fresher, you go into the lab. So we don't know so how to use this, because in colleges we are simply just theoretical part only we know. So after entering into the any pharmaceutical industry for as a fresher, then keep on going. Oh yeah, this is HPLC, and this is gas chromatography, and this is LCMS. and this is XRD. So like that, so after we knowing the instrument, so how to apply this instrument for our compounds. So these are all the techniques and applications. So for HPLC, this separation of compounds, qualitative and quantitative, and gas chromatography, analysis of volatile components, and LCMS, high performance liquid chromatography, hyphenated to mass spectroscopy for impurity profiling, characterization, and powder X-ray diffractometer is used for polymorphism, pseudo polymorphism, and patent productions, and identification and characterization of our synthesized compounds. So these are all the techniques presently using. So DSC, differential scanning calorimeter for drug development, drug excipient interaction, how the excipients interacting with the drug. So these are, we can scan by differential scanning calorimeter, and particle size analyzer, Fourier transform infrared spectroscopy for identification, characterization, and polymorphism. And AA, atomic absorption spectroscopy, inductor coupled plasma, and ionic chromatography, capillary electrophorese. This capillary electrophorese we can use for both organic and inorganic compounds. For ionic chromatography is especially used for ionic compounds to separate. And this atomic absorption spectroscopy and ICP also we used for elemental analysis. So these are the applications. So of course it is a vast subject because we don't cover in this session all the instruments. So right now just I am covering only HPLC, GC, and mass spectroscopy. So if you go this slide, high performance liquid chromatography, can I yeah, I asked some of the students have any idea about the high performance liquid chromatography? Is there any analytical students? Is it audible?
Yes,
Is
yes,
it,
sir.
Jyoti ma'am,
Yes, sir.
what are the courses you are offered in your college? That is analytical in chemistry?
Chemistry, only organic chemistry, sir, in my college.
Oh, there is
So
no
universities, analytical chemistry is there.
Okay, okay. So is there any students from analytical chemistry?
No, sir, no.
No, no.
Only organic background, sir.
Okay, okay, okay. So organic means okay. So I think they don't have that much awareness. Okay. Okay. Yeah. Okay. Then, so I will explain briefly about the high performance liquid chromatography. This is a very simple principle. What is this high performance liquid chromatography is? It's a schematic diagram I shown here. Very simple. We use some mobile phases and one column, and for injecting our sample, some sample components. What is the simple mechanism here is you use the mobile phase and you use the column and you inject the sample. So what happened? So what is the separation technique going on? A simple thing here. The stationary, the analyte sample, it will move between a mobile phase and a packing material of the column. So whenever sample enter into the column through the mobile phase, why it we call is liquid? Because we are using here liquid mobile phases. So that's why it is called as liquid chromatography. So why it became high performance? Because we are applying here some pumps which are very sophisticated pumps to pumping that mobile phase through the column along with the sample. So once a sample enter into the column, depending upon the chemical structure of the analyte, the molecules are retired while passing the stationary phase. So yeah, for reading it is very simple, no? But
Yes.
actually, this in industry, the HPLC we are using for so many products. This simple theoretical is there, but so much vast applications is applicable by this simple principle. So if you see the schematic, this is the solvent reservoirs. Uh, uh, Nirmala Madam ji,
Yes, sir.
you have any tours by our students, any college tours or to any labs? No?
HPLC is not there, sir.
Okay, okay. No, not in college, madam. You have any recent tours taking students to nearby labs, any
Yes,
pharmaceutical
sir.
company?
Yes.
No. Okay. So,
In Guntur, one lab is there, sir.
okay, your students are visited anytime?
Oh, sir. This year visited, sir, one time. Huh?
Okay. So, if you go through the HPLC, they are usually these are the modes we are using in HPLC: normal phase and reverse phase. So, what is the normal phase? So normal phase is I told you, no, one it is mobile phase and one is stationary phase. So here normal phase is our stationary phase is the polar and our solvents is the non-polar. That means mobile phase which is you are using is the non-polar solvent. That we call as normal phase. So we are using polar stationary phase like silica, cyanopropyl cellule, phenyl, amino. So the column we are using the silica and cyanopropyl cellule groups, phenyl, amino, by manufacturing the column by this chemistry, then we call it as a polar stationary phase. So, and our mobile phase is non-solvent because non-polar solvent. We are using NHane, NF10. So by using that non-polar solvent and using polar stationary phase, then we call it as normal phase mode. So usually in pharmaceutical industry, this normal phase we are using to develop the methods for chiral separations. So normal phase mode is very, very useful and very important because to chiral separation is very, very important in the pharmaceutical for the drug. Is there any chiral center? So definitely we have, because the chiral center is also one of the impurity for because you know that specific optical rotation is also very important for a drug. Whether the drug is negative effects and positive effects, if it is in chiral center, it is in maybe that drug is effective for one chiral center and it is negative for one chiral center. So the here one that impurity is also chirality is the one of the impurity for a drug. So to overcome how much impurity that is there, so normal phase method we develop and we give the method for developing the normal for the drug chirality. So normal phase mode is also very important and more useful. Wait a minute. Okay, is it okay now? Is it okay now? Okay.
No problem.
So, yeah. So if you go through the reverse phase, here we are using the stationary phase is non-polar and our mobile phase is polar. So octyl cellule, octadecyl cellule, this is the column chemistry we are used as a stationary phase. And polar solvents we are using water, methanol, acetonitrile, tetrahydrofuran. These are the polar solvents we are using. So many organic compounds. This river phase technique mode is very useful because so many HPLC methods is reverse phase only. So if you see so many papers published on HPLC, that almost reverse phase methods is very useful mode, particularly in the pharmaceutical industry. So many organic compounds we can easily separated by this mode. So when you see the structure of the high performance liquid chromatography, So I told you know that stationary phase, which is called as column, that called as solid support. So what is that column? How it works? Actually, in column we are used the silica, which is chemically bonded and with a particle size of 10 micrometer, 5 micrometer, 3.5 microns, 3 microns, 2.5 microns, 1.5, 1.7 microns. So uh, the particle size that is solid support, what the particles, what is the length of the column, what is the particle size. So depending upon the length of the column and particle size used, our separation is very, if you use very low particle size, that separation is very good because we get sharp peaks and resolution is good. So because that depends upon the method. Once here I am not more stressed about the methods because all students know, just they know my topic here also only just presenting the techniques. When you go the methods, that is separate subject. We can discuss later in some other any webinar. So right now I'm just discussing what is simple, what are the columns and what are the mobile phases, what is the general principle used in the HPLC. So, so that is the solid support we are using in the stationary phase. So bonded phase, functional groups firmly linked with to the solid support. Usually we are using column C8, C18, C4, etc. So for the columns, we are using some guard columns also because to protect the column from interference from the mobile phases and from the sample. So we are using some guard columns also. Yeah. So next detector. So one of the part in HPLC, one mobile phase, stationary phase, and finally detector. So because after separating from the column, it will observed by the detector. It is single wavelength or variable wavelength and multiple wavelength. So UV absorption here. So here some different detectors we use depend upon the method you develop and depend upon the moiety, analytical moiety you are used, that sample moieties you are using. So fluorescence detector, electrochemical detector, mass spectroscopy, refractive index, evaporate light scattering, so chiral detectors. So these are the detectors we are using in HPLC, which are suitable. If you go through the chart, so here I given chart here. Once here, if you see the drugs, favipiravir, molnupiravir, remdesivir, ritonavir, and ivermectin. So these are the drugs now very familiar because of corona, because these antiviral drugs presently using for curing the corona to some extent. Earlier we now I'm also working on molnupiravir, which is an antiviral drug uh, to cure the coronavirus to some extent. So this is the chart which I getting from my some of the products. So see by HPLs how they are separated, and first one is favipiravir, molnupiravir, and remdesivir, ritonavir, and ivermectin. So these are the retention time and concentration, how we use resolution and asymmetry. So this is the simple chart for HPLC. Yeah. So here technology wise, we used improve our technology from HPLC to UPLC. What is HPLC and what is UPLC? Here UPLC is nothing but ultra high pressure. We applied usually some extent we can go for HPLC because HPLC is 400 bar, more than 600 bar we can't go for HPLC. So why here pressure is also very important. Because of the pressure we can separate our cost effectiveness will, because our time saving, because usually HPLC is normally methods taking 1 hour, almost 40 to 60 minutes it will take. So what we have to do to time so much consumption is going on. So to overcome the time, consumption by the instruments. So one technology introduced, that is innovation UPLC. What is that UPLC? Ultra high pressure liquid chromatography. So we applying more pressure to HPLC. So due to applying the more pressure, the impurities may be separated in HPLC in around 60 minutes. You can separate these all in within 10 to 15 minutes also. So how much time you can save by this instrument? So almost 30 minutes, 30 to 40 minutes, we can reduce our time in every single analysis. So like this, so this is the technology. So analytical, what is the technology here is, so from HPLC now we are using, we are transfer our methods to UPLC by what is the, by using a ultra high performance liquid chromatography. What are the advantages? Faster analysis, peak to peak resolution is improved, high sensitivity and better signal by noise ratio, savings of time and cost per analysis. So why we use HPLC in pharmaceutical industry? See, when scientists synthesize the drugs, so they can know. So yeah, this I synthesized one drug related to this for any disease. This drug is suitable for the disease, though they synthesized in the synthetic laboratory. So then how they know this their drug is pure? So they can take analytical support. How my so what I synthesized my drug, is it correct? What are the characteristic properties of my drug? What are the impurities available in the drug? So because during the synthesis process, they used some chemicals, reagents, some reactions they used. As an organic chemistry students, you know all, for moving the reaction, you have to add reagents. So you have to add some chemicals, which are maybe at it is very adverse effect sometimes. So that all we are clear, and finally we have only quality product. So scientist will take help the analytical scientist. So how this is my drug. So what is the exact now drug substance? Is it the really good quality, or any some impurities are available? So we, based on the drug and impurities available from the, what is the scheme available from the synthesis team, we taken that synthesis, that scheme of the synthesis, how this drug synthesized. So we take the scheme and what are the possible impurities so we can expect from this scheme. So we take all the data from the synthesis scientist and we evaluate our data by developing method through HPLC. So how the drug, because it is a chromophore is there for the drugs and impurities. So what are the analytical instruments? Is it suitable to develop this method? So we as an analytical scientist, we do some work, research work about that, how to develop for a method for this impurities, and for this, what is the instrument it is good for this analysis. So we do some research by some taking some articles about the drug, because what are the publications about the drug. So we see some analytical research work and we done some work and we do development according to that development. So this development gives us feedback how this drug, so how much impurities available, so how much percentage it is there in that drug. So because why it is important? Because you are just simply synthesize the compound is not, because that is not a completion of work. After the synthesis of work, depending upon the analytical data, you go for registration. So during this process, that regulatory bodies ask how much low level your drug has impurities. So this is the slide I am showing here. As per ICH, any new drug if you develop, that impurities level should be should be level 0.1% threshold level for if your drug is taking intake 1 gram or less than 1 gram. So for suppose if it is greater than 1 gram in your drug quantity intake, so then its impurity should be 0.05% level. So what are the regulatory bodies? They given some data. So when you apply for any registration for drug, so these are the maximum daily dosage, how much threshold that impurities have. So you have to follow that regulatory guidelines. When you are going to registration, you have to follow the guidelines. So the synthesis scientist given the drug, and we maximum tried to develop our methods to identify the impurity level below that guideline levels. So we have to develop the methods by researching the work. So how to develop, what is the observance of that moiety. So based on, see, that's why here analytical, why it is very important in pharmaceutical industry in any analytical team, because simply synthesis is not the completion of the project. So you have to synthesis, and we have to give the analytical support, good quality product we have to get. So we have to do our side analytical support. So HPLC, by the HPLC we can detect the impurities, organic impurities. So next we go through the another technique, very important technique. This also gas chromatography. Yeah, simple. See, in HPLC, that is high performance liquid chromatography, and this is gas chromatography. So simple, gas and their liquid. So what is the difference between, by wording itself, it shows the difference. Liquid chromatography means that you are using mobile phase, and that is liquid phase, and here the mobile phase you are using is gas. So here also a simple principle which is based on the partition of the analyte between the gas and a thin layer of non-volatile liquid held on the solid support. So here also one stationary phase we use, there we call is column, here also we call it as column. But here some capillary columns we are used, which are having solid support in that. So what is the applications of this gas chromatography? So this schematic diagram, if you see gas chromatography, here the mobile phase is gas, and here there is a oven, and with column you are connected, and some detector at last. From the detector we get the response, how it is responded, if you see in the computer. So see here mobile phase is carrier gas. Usually we are use the mobile phase gases here is nitrogen, helium, or hydrogen. Why particularly these gases we use? Because these are inert in nature, and there is no interference, because if you are analyzing any volatile, so if any some interference in the mobile phase, that what are the analysis you done is not correct. So that mobile, so there is no other than moiety, there is no such interference from any other the component. So that's why we have to use mobile phase, which is inert, chemically inert, and this is very highly pure. We use 99.99% purity carrier gas we can use. And this packed columns usually used for conventional low sensitive GC analysis, and capillary. Here two types of columns we are used regularly in GC analysis. One is packed column and one is capillary column. So for packed column, we are used conventional low sensitivity GC analysis, and capillary column is a new technology column used for better sensitivity, generally with headspace GC analysis. Here in GC also we have, for sample, we have two modes, direct methods and headspace method. So we use, by using these two methods, we're developing the methods. So like this tubing, column material, stationary phase, film thickness. I earlier said in HPLC, the particle size. Here we are call it as film thickness. So how much film thickness it is? 1.8, 1.4. So some measurements is there. Yeah, so this is the stationary phases. Generally we are used in gas chromatography. 100% dimethyl polosiloxane, diphenyl dimethyl polysiloxane, cyanopropyl phenyl dimethyl polysiloxane, trifluoropropyl dimethyl polysiloxane, and polyethylene glycol or wax. So these are the column chemistry which are generally we are using as stationary phases in gas chromatography. So depending upon the method when you are developing time, so what are the residual solvents? Because gas chromatography is familiar, because what are the solvents at the time of synthesis in the product? So we have to identify the solvents, how much level, how much ppm level it is present in your drug. So here also, as per guidelines, how much residual solvent must be should avoid. So these are the, so by developing the method, we go that much sensitivity level, and we give the data according to, so how much this is the residual solvents present in your drug. So this all we have to give by GC chromatography. So see, this is the one of the chart for GC analysis chart. If you see, this is methanol, acetonitrile, methylene chloride, trans 1,2-dichloroethylene, cis 1,2-dichloroethylene. So these are the peaks for that residual solvents. This is a simple chart for GC chromatography. So many solvents, especially for GC chromatography, you use it for identification of solvents in pharmaceutical industry. See why, as per like HPLC in impurities in a new drug substance, like what are the residual solvents when the synthesis time, what are they are used residual solvents. So usually guidelines differentiate the residual solvents as a 4 category right now. So if you go through class 1, it is highly toxicities, because this type of solvents almost, if you avoid in the synthesis, at the time of synthesizing the drug, you have to avoid this residual solvent. You have to avoid this solvent, which is belongs to class 1. If you class 2, that solvents belong to class 2 is medium toxicities, and class 3 is low toxicity. So when scientists at the synthesis time, here I have to take care of this, what class solvent I am using. So is it okay for using this solvent? Because later on, if you are used any class 1 solvent, knowing or unknowing, if you use any class 1 solvent, later on at the time of analytical stage, we told why this much use class 1 solvent you use. So that means your drug is failed. So later on, regulatory bodies also no way they reject at the time why you use a class 1 solvent. So when your synthesis time also, scientists are very care about this class about the solvents, which class they are belong. Is it okay when we're using for the synthesis? So they take care of this solvents also. So if you go through the class 1 solvents, it is benzene, carbon tetrachloride, 1,2-dichloroethane, 1,1-dichloroethane, 1,1-trichloroethane. These are very, very dangerous. We completely avoid these solvents, because if you see benzene, of course, if you use also, because completely we avoid is a very, very good option. Maybe if they use also, regulatory agencies given the concentration limit 2 ppm for benzene, because benzene has adverse side effects, like it is carcinogen. When you use benzene, it is leads to the carcinogen. So carbon tetrachloride, it is toxic and environmental hazard. So if you use carbon tetrachloride, it gives very toxic and adverse effects to the environment. So why you are using when you our synthesis time, why you are using these solvents? So 1,2-dichloroethane, this is toxic, and 1,2-1,1-dichloroethane, this is also toxic, and 1,1-trichloroethane, environment hazard. So the scientists have to take care of that before doing the synthesis. So what are the solvents we are say when the reaction time, what are the solvents we can available useful, because goes on because every step we have to take care of, you have to take care of every step. So because before only if you reduce the safety precautions you are taking, so the at last we get very good quality product. So by stepwise we have to regulate all these things. So these are the class 2 solvents. Regularly this acetonitrile, chlorobenzene, chloroform, cyclohexane, 1,2-dichloroethane, dichloromethane, dimethyl estimate, and methanol. So usually acetonitrile, methanol we are using regularly. So see, these are also class 2 solvent. So that means okay, it is medium toxicity, but toxicity is there. So usually people in the labs and all in organic synthesis time in lab, and we are generally take what acetonitrile methanol. They're simply it is so much adverse effects is there, but people are thinking, oh, methanol acetonitrile, it's okay like that, but it's under class 2 solvents. So you have to take care about of this acetonitrile and methanol also. See, for acetonitrile, see 410 ppm is the concentration limit. So see how much less it is there. But simply people, students are simply exposed to acetonitrile and methanol. So take care of this. If you go for class 3 solvent, that means a very little toxicity. This acetic acid, acetone, anisole, cumin. So these all come under class 3 solvents. And one more category is that is class 4 solvents. These classes of solvents are those for which no adequate toxicology data, because these are not under, because they don't have any toxicology data. So because regulatory bodies are doing every time they getting the data, what are the effects of toxicology, they get toxicology data. Based on that data, they evaluate the solvent comes under which list. So if you kept any solvent in the class 4, then it has no that much adverse effects. So there is no toxicology data about that solvent. So at the synthesis time, this class 4 solvents and class 3 solvents, we can easily use it without any limitations. Yeah. So next, go to the other technique, that is mass spectroscopy. So mass spectroscopy, what is mass spectroscopy? Mass spectroscopy is very superior scientific tool nowadays, because to quantify and identification of unknown compounds and known compounds and structure elucidation. So you synthesize the drug, if a scientist synthesizes the drug. So at the time of the drug, we are doing the analysis, some unknown impurities also will come, and some degradation studies time, some unknown impurities will come. So how to identify this unknown compounds? So by this scientific tool, mass spectroscopy, we can easily identify it. So right now, presently, we are using mass spectroscopy instruments in pharmaceutical industries, single quad LCMS, tandem LCMS-MS, and GCMS, time of flight. So these are the recently now we are using mass spectroscopy instruments. See, this is the schematic diagram of this. Yeah, this is very simple. If you see what, if for organic students, already very familiar about mass spectroscopy, and I think spectroscopy, all mass spectroscopy and NMR is very familiar, I think, because theoretically very good in PG level. It's very useful in at the time of job. The theoretical part is very important. If you see that simple, that the test sample what you introduce simply into the mass spectro instrument, its electric beam bombarded that sample moiety into the positively charged repeaters and negatively charged replicate. So you completely in that instrument, the complete vacuum, you bombarded that moiety, so it distributes like m by z values. So at the usage of that magnetic, that analyzer tube, this positively charged ions and they are separated, and due to that how the fragmentation happened, based on that fragmentation, that molecules move to the detector level. There we identified based on the m by z. So we calculated according, so by that values. What are the main components in mass spectroscopy? Inlet system. So because how you introduce the sample in the mass spectroscopy? By LC, liquid chromatography you can introduce. By gas chromatography you can introduce. By direct probe, that means you can give sample directly to the, without any using HPLC and GC, we can directly inject the sample also. That is direct probe. So what are the ion source used in mass spectroscopy? EI, electron ionization, CI, chemical ionization, electron spray ionization, and MALDI, APCA, etc. So what are the mass analyzers? How you analyze that ambijets? By quadrupole, time of flight, ion trap, and magnetic sector. So what are the detectors used in mass spectroscopy? Electron multiplier, microchannel plates. So these are the main components. We go through the mass spectroscopy. Let's see what are the applications. So, but very nowadays mass spectroscopy has very it's very important in the pharmaceutical industry, because at final stage, due to mass spectroscopy only we can more and more we can increase the quality of the product, because by mass spectroscopy we can identify uh, every regulatory bodies asking what are the mutagenic or carcinogenic impurities. At the synthesis time they have little doubt about that impurity. Want to ask the complete characterization of that impurity by using mass spectroscopy, NMR. So complete data, complete characterization, how the moiety, is it is any carcinogenic or it is any toxicology data is available. So the based on the work and what is the analytical data you are giving, that very, if you use any moiety which is leads to the toxic and carcinogenic, maybe little they have some little effect with that impurity also. They want full data how you analyze that impurity. So by this mass spectroscopy and this NMR spectroscopy, we can easily identify at very low level, and that impurity, maybe if you are present in the drug, that impurity is very less quantity what we synthesized by our product. So my product is good. So by the mass spectroscopy and NMR data, we completely evaluate the impurities at very low level, because of the sensitivity and accuracy of the instruments. So if you simple go through the chart, so this is a chart of the mass spectroscopy and relative intensity and ambiject value. So here see 29, ambiject value 39, 43, 55, 83, 98. So simply like chart we will get like this. If after fragmentation, the chart will show like this. So based on the calculation, so you have to identify. You already here only theoretical application will theoretically we know so how the fragmented happen. Theoretically we actually calculated, so this fragmentation happen means this molecular weight will come like that. But instrument itself it give all the data, so you are simply just by collecting the all data you can simply tell, oh, this is a molecule structure of this impurity like that. Yeah, as a conclusion part, so technological advances are being happened in pharmaceutical analysis like other areas of science. So many advances happening year on basis. So due to technology advances, more and more pure drug will be available for patients, and drug testing becomes more regulated. So I earlier said more regulated means you have to go more work we have to done to reduce the impurity levels at very low level. To patient, as we take care of the patient usage, patient how much it is using the drug daily. So based on that, regulatory bodies are giving, so this much level is only this much level is okay for, but remaining you have to do so much work. So with the new technology, instrument and automation, productivity is drastically increased, and hence it benefits the business of the company and also to delivering quality product. If you see in this pandemic situation, so how we get this vaccines, because due to this technology, instrument and automation, so much productivity. So we supplied, if you see in India, so much we supplied for other countries also because of the automation and technology instrument. Because without this instruments and automation, we can't product that much quantity. So now we overcome within 2-3 years this virus, means due to this technology instruments and automation, now installed in the pharmaceutical industry. So with the support of quality analytical data, companies are able to protect their innovations by patenting them. So earlier I show some XRD instrument. So how XRD is useful means, so because of this polymorphism, so many companies they are applying for any patent. So based on polymorphism, so they can what are the innovations they've done, they can patent them by the polymorphism of the molecules. So by little change of polymorphism also we can patent the molecules. So yeah, I'm concluding my session from here, and thank you.
Thank you, sir. Any queries, please ask. Sir is ready to give the answers.
If you go messages,
Sir,
oh,
how can NMR be useful for coordination complexes? One more question from participants, sir.
how can
NMR useful for coordination complexes.
yeah. What coordination complex means? What, madam? What particular? What is the coordination complexes?
I don't know, sir. Coordination complexes.
No, usually in pharmaceutical industry, what we do, what are the drug that they are synthesized. So we characterize that drug by analysis through a nuclear magnetic resonance. So what is this coordination complexes? So how an actual particular, I want that particular terminology, because we are regularly using in industry. Our motto is like how synthesis, that synthesis part, what the synthesis the drug is, according to that we give the analytical data. So we do the analysis NMR to characterize that particular drug only. So here terminology, what is the coordination complex? I want that terminology. So you want to the drug or something, what?
మేడం ఆగండి మేడం, ఒక నిమిషం ఆగండి.
One more question, sir.
Yeah.
Kindly discuss how to evaluate NMR for complexes.
Yeah, just now you told me, no, ma'am.
Yes, yes.
What are the complexes I want? Because we are simply doing the drug according to the drug part. What are the, if any synthesis time, we are analyze the drug. So particularly the drug, we give the characterization of the impurities. Particularly we give the we analyze through NMR, nuclear magnetic resonance. We give the characteristics, because synthesis people, they know the synthesis, but particularly that molecular weight, that that's related structure is there or not, they don't know at that time. So by NMR we can characterize that drug. Yeah, your what are the molecule you synthesize is correct.
Sir, one more question, sir.
Madam.
Sir, stationary phase is available or not?
Which one, madam?
Stationary phase is available or not?
What stationary phase?
Stationary phase.
Yeah, madam. What stationary phase? What?
I don't know, sir. One more.
No. Stationary phase, usually stationary phase in HPLC. If you go HPLC, high performance liquid chromatography, here stationary phase all manufacturing, madam, because we are simply use columns. That's why in theoretically and practically so much difference is there. Theoretically we saw we at the time of theoretical time, because if you go industry all available. What is the stationary phase here means here one column just we
Yes, yes.
connect the column. What is the stationary phase? Theoretical here what you understand is that column stationary phase, how column chemistry is working.
Yes.
Here theoretical inside is they already manufactured one column. So what is the chemistry involved in this column? So when we developing the method, that time this column chemistry is suitable for my method. So we select the stationary phase like that. So whether my drug, my compounds, my organic compounds, which I have to evaluate, if that stationary phase is suitable. That means whether I use a C18 stationary phase, C8 stationary phase, amino phenyl groups. So what group I can use for separation of these compounds. So stationary phase is available or not, that is not, actual stationary phase is available. Without stationary phase, there is no analysis.
Anyone, please ask. Anyone? Sir is ready to clarify your doubts.
No, Nirmala madam, what
Madam,
about
sir?
the lecturers who are working in chemistry department? They have any doubts?
So many are there, sir.
Yeah, you can, no problem. Because I am also a background from organic chemistry
Organic
in
chemistry.
masters, but I changed. Fortunately, maybe
Yes.
unfortunately, I don't know. But fortunately, I can think because analytical chemistry is no much exposure to chemicals compared
Yes,
to
yes,
organic
yes.
chemistry.
Organic chemistry.
Yeah.
That's the drawbacks.
So we are very little quantity we are used just for analysis purpose.
Yes, yes.
So we don't have that much exposure to chemicals, but synthesis people have so much exposure.
So
So their lifespan.
analytical chemistry in Acharya Nagarjuna, sir.
Okay.
So all are in organic chemistry background, sir.
Okay.
Mm.
Saujanya has put one question. Sir, stationary phase is available or not?
Sir already explained, sister.
Explained, okay.
I explained
Yeah.
already.
Morning, sir.
Uh,
Myself, Dr. Sarojini, Zoology Department, Pharmaceutical College.
what? Madam, your voice is not clear.
Sarojini madam, your
Type
voice
it,
is very
Sarojini.
low.
Type it, Sarojini. Voice is not clear.
Yes,
Type in the chat box.
chat box.
Myself, Dr. Sarojini,
Oh, okay, ma'am.
Railway College for Women, Tenali.
Okay.
I'm following your lecture very carefully.
Okay.
You told some of the chemicals are cancerous, carcinogenic.
Yes.
Is it good to use these perfumes, all those ones?
Madam,
No,
your
madam.
voice is very low, madam. Sarojini madam.
Using
Please raise your voice.
using perfumes, is it good?
No.
Using perfumes is good or not?
Using
Perfumes.
using perfumes is good or not? See,
Yes.
madam, yeah, actually, your question is very good. See, ma'am, because as a pharmaceutical industry, we are using in perfumes or any chemical industry what they are manufacturing, definitely they use chemicals. Chemical itself is having some adverse effects. There is no doubt in that. But here, what you said here is, for suppose in a human, if you take any disease, if he has cancer, so here, if you take medicine, his lifespan will increase. If you not take medicine, he will die. So here, we have take medicine to decrease, to you have to use medicine to save his life from cancer. So here main motto is that. So for that purpose, here for synthesizing of that medicine, we have used some chemicals. So, but based on that, your side effects. So how much effect, because your disease is going, you are reducing by disease by using this medicine, but little side effects is definitely there. So every chemical product have some, because that's why from pharmacovigilance is there, one team is there. They keep on working on this. What are the side effects? If every industry has some pharmacovigilance team, they do what they work. Once you deliver your drug, so how many patients you used your drug, any data will came from that. So any side effect. So based on the data, every time we are changing our quality of the product by taking the feedback of the patients. So like perfume also, if you use perfumes to avoid, if you avoid means that is okay. But here by using perfumes, you have some definitely some solvents is there. We are using some solvents, so it will have some adverse effects. So perfumes using or not using, that up to your level.
Yes.
Of course, but nowadays we are trying to enlighten our students because we have studied, while we are studying the organic chemistry, aromatic compounds are cancerous. That's why I used to just ask the students not to use much perfumes, even my kids. But
Yes, madam. Correct.
uh, chemicals, that also powdery
Yeah,
material.
that's why, madam. That's why, madam. Because see, at the industry level, all for suppose if you go for perfume industry, worldwide, some they are also have some regulations. How much level you have to use to synthesize your product? What are the impurities? So what are the solvents if you are used? So how much level it is? Because once you submit any application for the agency, they go through all the what are they follow the all regulatory guidelines or not. Once they are all follow the guideline and your product is satisfying all the guidelines, then only they can approve. So uh, so worldwide, all industry have some guidelines, especially for chemical industry. There's so much more regulations compared to other industry.
Thank you.
Okay, madam. It's my pleasure.
If there are no questions, then we can.
Boledu Suchita, very informative session. Thank you, ma'am. On skin, it is not good using.
Perfume.
Yeah, perfumes. Yeah, correct.
Yes.
Not analytical chem. Kindly discuss how to avoid. Yes. Okay, now.
Okay, I think we can conclude because there are no questions further.
Okay, yes. Yeah, if you have lecturers have any questions also, I am ready to answer because they are not aware of much about the industry. Because the lecturer will know very well, so students automatically will know well. Because they are the inspiration for us. So once lecturer will give, because at my time also so many lecturers inspired us because of their teaching subject only. Oh, okay. So because we are proud as a chemistry student because we are saving life. Because what you think all chemistry students, they are actually, we are thinking, yeah, software people are very, they are good salary they are getting, but as a humanitarian grounds, chemistry students are very proud to say that you are saving the lives. It is
Yes, sir. Thank
humanity
you,
is
sir.
there
Thank
in
you.
chemistry. Humanity is there, madam, because now in corona time, who are front warriors?
Yes, sir. Yes, sir.
Yeah, our chemical
It's
field
true, sir.
only. Yeah. So you are proud of as a chemistry student, and you do well, and very happy you are become future scientist.
Yes.
So please serve the country and please serve the humanity.
Sure, sir. Sure.
And very thankful to the management to giving this opportunity, to the
Thank you so
principal.
much.
I don't know your name, ma'am. What's your principal, ma'am?
Sister
Sister
Shiny.
Shiny.
Shushai.
Sister
Shushai.
Shiny.
Shiny. Thank you,
Yes.
ma'am. Shiny, ma'am. Yeah.
Thank you, sir, sharing your rich knowledge and the very availability that you
Oh, thank
accepted
you.
to share your
Thank
rich
you.
knowledge. Thank you, sir.
Thank you.
Really, God opens knowledge to few people to serve all the other people. Okay. So it is nice to hear you, sir, and
Thank you, ma'am.
very happy
Thank you.
the knowledge that is shared by you.
Yeah, my one sincere suggestion to the lecturers and college management team, please bring the students some like tour, madam, because
Yes, sir.
when you
Field
visit the
trips.
industry,
Okay.
really, okay, this is the, because earlier that much no awareness, because we are simply just reading the books. Because once theoretically it is good, when it is good, after you know the instrument, what is happening, that time theoretically okay. So theoretically it is here applicable here like that. So sometimes tours you are arranging means the students are aware, okay. So industry is like, because if you at the student time, if you are aware of the instruments, you can easily involved in the work. Otherwise, for 1 year, 2 years, you for learning the instruments only it will go. But okay, that and all it is okay. It's a part. It will go. But before awareness is very far better than after entering into the industries. My suggestion is take some tours. It is available. It's
So,
after the
due to
management team.
Corona, sir, due to Corona,
Yeah,
we are not willing to visit the industries, sir.
yeah,
That's
yeah,
it.
yeah, madam. Yeah, yeah. But that is industrial tour is very because even so many universities now they are following that industrial tours and
As a
yeah. But
part
this
of
is there in early. Earlier is also there because at the time of I am schooling, my model high school in Rajahmundry, AP Paper Mill Model High School, they taken me at the 8 plus. They taken to Nagarjuna Fertilizers that time.
Yes. Okay, okay.
Yeah, feel happy that one like tour.
actually, sir, as a part of curriculum, every year we take the students for field trips and study tours. Only
Okay,
this
okay.
2 years, past 2 years, due to Corona pandemic, we have not
Yeah.
taken. Otherwise,
Yeah. That's true, madam. Because now all colleges and curriculum they are adding this. Yeah. But just like one more, just it's a
Okay,
suggestion.
sir. Sure, sure.
thank you, sir.
Thank you, Dr. Raja Garu, for your wonderful and excellent presentation on HPLC, column chromatography, gas chromatography, and its application in pharmaceutical industry. Thank you, sir. Thank you once again. Now I hand over to G. Saujanya to present a vote of thanks.
I feel
I feel
honored
honored
and
and
privileged
privileged
to
to
propose
propose
vote of
vote of
thanks
thanks
on this
on this
special
special
occasion.
occasion.
I
I
would
would
like
like
to
to
thank
thank Dr. Raja Gorla, Senior Scientist, Analytical Research Department. Senior Scientist, Analytical Research Department, Donbang Life Science Limited, South Korea. Sir, your session on advanced techniques and applications in pharmaceuticals industries to achieve quality products is very informative and it analyzed in various aspects like instruments have revolutionized chemical analysis, replace what chemistry methods and detailed explanation HPLC, gas chromatography, thin layer chromatography and mass spectrometry and ultra high performance chromatography. Thank you, sir.
Now I hand over to him.
So I take this opportunity once again on behalf of the college family and the Department of Chemistry. I wholeheartedly thank Dr. Susan Kumar and Dr. Rajagaru for your very availability to share your rich knowledge with with the teaching fraternity. We are very happy, and the participants' response itself we can see they are giving very good response of your expertise and the knowledge that you have shared to the community. And I am sure that it will be great help and useful to the students as well as the teaching community. So once again, I thank you and I congratulate the Department of Chemistry, Dr. K. Nirmala Jyoti, the convener and the co-convener, and all the faculty members of Chemistry Department for organizing this 3-day international webinar so successfully, fruitfully, and productively. Thank you. May God bless you all. Thank you, sir. Thank you. Thanks.
Now I request Mrs. Parimala Jyoti to continue the next session. To present the consolidated report of these 3 days webinar.
Thank you, Madam. I feel it a privilege to present the concluding remarks of the 3-day international webinar organized by Department of Chemistry, JMJ College for Women, Tenali, on the topic Recent Innovations in Pharmaceutical and Applied Sciences. Dr. Dasari Ayodhya, Examiner of Patents, Designs, Ministry of Commerce and Industry, Tamil Nadu, Government of India, was the resource person for first day of the webinar. He delivered a lecture on introduction of nanomaterials, conversion of bulk materials into nanomaterials, types and examples of nanomaterials, different types of synthetic methods, characterization techniques, properties of nanomaterials, and finally, various applications of nanomaterials. Professor Ratnakaram Venkatnath, Professor, Chemistry Department, Deputy Director, Admissions and Research Coordinator, Geetam Deemit to be University, Bengaluru, Karnataka, India, was the resource person for second day of the webinar. He delivered a lecture on chemical, biochemical, and microbial advances in process industries. During his representation, he explained about innovation, contribution of GDP in India, how this digitalization is going to transform the entire society to improve the process industry, ways to pharmaceutical companies are using data analysis to drive innovation and ultrasonics. Dr. Susan Kumar Vadagoppala, physical therapist, dynamic balance physiotherapy, Vancouver, Canada, was the resource person for session 1 of the 3rd day of the webinar. He delivered a lecture on lifestyle modifications for better health and positive well-being. During his presentation, he explained population, public versus private healthcare, health, heart disease risk factors, habits, and blue zones. Dr. Raja Gorla, senior scientist, Dongbang FTL Limited, South Korea, was the resource person for session 2 of the 3rd day of the webinar. He delivered a lecture on advanced techniques and applications in pharmaceutical industries to achieve quality products. During his presentation, he clearly explained about HPLC, gas chromatography, TLC, and mass spectrometry. Now I would like to invite Dr. K. Nirmala Jyoti, HOD of Chemistry and convener of this 3-day international webinar on recent innovations in pharmaceutical and applied sciences. Thank you, one and all.
Yeah.
Nirmal Jyoti not able to hear. Please
మ్యూట్ లో ఉన్నారు అన్మ్యూట్ చేసుకోండి.
Okay.
the microphone.
A warm welcome to one and all. It's my great privilege and honor to propose a vote of thanks speech and acknowledge the contribution of those who worked really hard to make this event happen. Let me first of all start by giving glory to the Almighty God for making today's day a resounding success. My very special thanks to our correspondent, Dr. Sister Teresa Magade, and our beloved principal, Dr. Sister Shaini KP, for their valuable contribution and constant support and guidance which made this event grand success. I extend my heartfelt thanks to the resource person of the first day, Dr. Dasri Ayodhya, Examiner of Patents and Designs, Ministry of Commerce and Industry, Tamil Nadu, Government of India, for enhancing our understanding pertaining to the topic general synthesis, characterization, and applications of nanomaterials, and for the outstanding presentation, which left a huge impact on the people who attended the webinar. I am very happy to extend my sincere thanks to resource person of second year, Professor Ratnakaram Venkatnan, Professor, Chemistry Department, Director, Admissions and Research Coordinator, Geetham deemed to be University, Bangalore, Karnataka, for exposing the theory of topic chemical, biochemical, and microbial advances in process industries. It was indeed a great experience having you at our event, sir, for sharing your rich knowledge and providing valuable insights on the subject. I extend my deepest gratitude to the resource person of third day of first session, Dr. Susan Kumar Vadagoppala, PT Physical Therapist from Canada, Dynamic Balance Physiotherapy, Vancouver, Canada, for not only sparing their invaluable time to grace this event, but also for enlightening us and providing a new exposure with their commendable talk on the subject, lifestyle modifications for better health and positive well-being. I am running short of words to express my humble thanks to the resource person of second session of third day, Dr. Raja Gorla, Senior Scientist, Dongbang, FTL Limited, South Korea, who is a source of inspiration and motivation for the research-oriented youth and providing his vast knowledge and acumen in the subject, advanced techniques and applications in pharmaceutical industry to achieve quality products. We are extremely thankful to Dr. K. Nirmal Jyoti, HOD of Department of Chemistry, convener of this webinar, for their minute-to-minute guidance and support and for providing encouragement at every point of time in the organization of this great event. I especially thank the members in the Department of Chemistry, Mrs. M. Shekina Kumari, Organizing Secretary, Dr. V. Sumalata, Ms. P. Parimala Jyoti, P. Kirtana, G. Saujanya, and Mrs. Y. Ramadevi for their tireless efforts in organizing the webinar and making it a great success. I wish to thank Mr. Murali and Mr. Ravi for providing technical support to this webinar session. Above all, I thank all the participants as your participation has made this webinar session a successful event. I believe that this lecture has certainly provided you an insight into the recent innovations in pharmaceutical and applied sciences. After this program, it is expected that whenever you will be dealing with it in your personal and professional life, this exposure will help you to formulate the policy with the angle of fair competition and great success. Once again, I thank you all for your attention, cooperation, and support. Thank you once again.
Okay, then I think we can disperse. So once again, thank you all. Thank you so much. Feedback will be sent to your mail ID. So you can.
Thank you, Susan Kumar garu, Raja garu. Thank you once again. Thank you, sir.
You're welcome.
Thank you, sir.
Thank you.
We will close the session.